Hi, I'm Janice Lee from Dr. Josephine's lab, and today I'm going to talk about how Neuregulin-1 improves brain endothelial barrier function after ischemic stress. So Neuregulin-1 is one of the growth factors that binds to ERB receptor and serves diverse roles in our body. But the role of this pathway in brain endothelial cells after ischemic stroke was relatively unexplored. So our lab decided to incorporate the in vitro ischemia model known as oxygen glucose deprivation (OGD) model to investigate its roles. After OGD and reoxy stress, the relative dextran and extravasation was increased, and this was decreased in the presence of neuregulin. Next, we wanted to check the level of tight junction proteins. However, under OGD and neuregulin treatment, the total level of these proteins nor the subcellular localization was not significantly changed. Next, we investigated the pathways relative to endocytosis. However, clathrin, MFSD2A, caveolin-1 levels were not changed under these conditions as well. Especially caveolin-1 is known to be regulated by neuregulin-1 or ERB2 receptor in different cell types. Therefore, we investigated how it is changed during these conditions, but they did not bind to ERB2 receptor, nor its expression or subcellular localization change. Next, we investigated the ectin cytoskeletal mechanism, and interestingly, after 30 minutes of reoxygenation, the phosphorylation level at serin 19 of MLC2, myosin light chain 2, was increased, and this phosphorylation was significantly reduced in the presence of neuregulin. Next, we checked the level of F-actin stress fibers, which is known to be controlled by the phosphorylation of MLC2. And we could see that the stress fibers were induced after OGD reoxy stress and decreased, somewhat rescued, in the presence of Neuregulin-1. Next, we checked the downstream of that pathway. We did not see the total level changes of adherence junction proteins. However, we could see that under the microscope, these proteins were retracted from membrane portions. This is the summary and conclusion. Neuregulin-1 treatment contributes to endothelial integrity by blocking MLC2 phosphorylation-mediated adherence junction disruption after ischemic stress. Thank you for your attention.